Hello everyone, welcome to the O-Level Biology series brought to you by ZNotes. I'm Mohammed Anian and I'll be your host. While I'll be your host, we have an amazing contributor, Nilanto Ahmed, who will be guiding you through all the key concepts and topics you need to know for O-Level Bio. Together, we'll make sure you're well prepared and confident for exams. Don't forget to hit subscribe so you don't miss any episodes. Let's get started and dive into the fascinating world of biology. I'm handing it over to Nilanto Ahmed now.
Good evening, everyone. I'm Nilanto Ahmed, and today we will be covering Chapter 4 of the Cambridge O-Level syllabus, which is Biological Molecules. We'll be covering the first five key concepts of this chapter, which are carbohydrates, lipids, proteins, DNA, which is a new subtopic that Cambridge has added in the 2023 syllabus, and also some food tests for alternative to practical and for paper 1 and paper 2 also. First, let's cover some key concepts for some of the subtopics. We will be covering again the carbohydrates, lipids, which consist of fats and oils, proteins, and DNA. We'll be also covering about how polysaccharides and many other macromolecules are formed from smaller molecules, which is only limited to starch, cellulose, and glycogen, from sugars, proteins, from amino acid, lipids, and DNA. Describe we have to also describe about some chemical tests for paper 2, paper 1, and the alternative to practical paper, which is paper 4, only limited to starch, sugars, proteins, and lipids. So first is carbohydrates. Now, what are carbohydrates? These are simple sugars, organic molecules that are made up of carbon, hydrogen, and oxygen, with the oxygen to hydrogen ratio being 2:1. These are basically found in all of the basic foods that we eat every day, like bread, meats, and milk. And carbohydrates consist of glucose, fructose, galactose, starch, and all the other types of sugars that are normally found in nature. Carbohydrates are of two types. First is monosaccharides, which are simple sugars that are composed of only one molecule, which bear the formula C6H12O6. In nature, they're crystalline and soluble, and they also have a sweet taste, such as glucose, fructose, and galactose. Polysaccharides are larger carbohydrates or complex sugars that are composed of many monosaccharides or simple sugars covalently bonded together. Unlike simple sugars, they are not crystalline in nature and are insoluble. Plus, they do not bear any sort of taste, such as starch, which are found in plants, cellulose, and glycogen, which are found in mammals. Now, what are the function of carbohydrates in living organisms? Carbohydrates require for respiration to release energy for various cellular processes, such as forming polypeptides and making more cell wall and other components. It is also used to form supporting structures such as cell wall, which is composed of cellulose. It is also converted to other organic compounds such as proteins. It is also used to produce nucleic acid for DNA and RNA. It is also and for producing glycoproteins such as mucus and to produce nectar in flowering plants to attract insects for pollination to occur. Now, you may come up with this question that why are glycogen and starch suitable for storage in cells? Because unlike simple sugars, these are insoluble, so they do not affect the overall concentration of cell cytoplasm. They are larger molecules, so they cannot easily penetrate and diffuse through the cell membrane. They can be easily hydrolyzed by enzymes to glucose, especially by the enzyme amylase. And unlike other types of components, these two occupy less space due to the shape of their molecules. Now, the next part we'll be covering is fats. These are also organic molecules like carbohydrates, except the only difference is that fats have less oxygen, unlike in carbohydrates. There are two types of fats depending on their state at room temperature. There is unsaturated fats, which we call Oils, these are liquid at room temperature, and they bear one or more carbon to carbon double bonds and are mostly obtained from plants. Saturated fats are mostly solid at room temperature, also called margarine, such as butter. They do not possess any carbon to carbon double bonds and are mostly obtained from animals. Now, what are the function of fats? Just like carbohydrates, they are also used to release more energy, but unlike carbohydrates, they're readily available source of energy, which means they can be easily broken down to release energy at a more larger amount. Fats also act as an insulating material to prevent heat loss. They're an essential part of the cell cytoplasm, and it also acts as a solvent for fat-soluble vitamins and some other substances such as vitamin A. They are used to produce certain hormones also, such as the five steroid hormones, testosterone, estrogen, and the other three reproductive hormones, FSH, LH, and progesterone. Proteins, these are organic molecules composed of carbon, hydrogen, oxygen, and nitrogen, and sometimes rarely sulfur. They are polymers of amino acids which bear an amine group, a carboxylic group, and a variable side chain, which may either have hydrogen or different acid groups. They're mostly found in both animals and plants. Food sources for proteins eggs, are eggs, meat such as chicken and beef, and milk. Proteins help to produce new cell cytoplasm. They're essential for growth, tissue repair, enzymes, hormones, and antibodies. Now, here are some tests for paper 4. It may also come up in paper 1 and paper 2 also. The first test is starch. To test starch, we use the reagent iodine. The procedure is we need to add only a few drops of iodine in a sample of the food. The positive result will be shown if the color changes to blue-black from brown. For sugars, we use the reagent Benedict's solution. To test for sugars, we first have to crush the sample in a test tube with water and add the same volume of the reagent. Like if for say I have 20 centimeter cube of the crushed sample in a test tube, then I will also add same amount of Benedict's reagent. Then we will shake and it in a warm water bath for some minutes at 80 to 90 degree centigrade. The positive result will be shown if the blue color turns to brick red. Plus, we can also determine the concentration of sugar in Benedict's reagent results. If there is low sugar, then the result may come up as green or close to red, such as orange. For fats, we use what is called an emulsion test. For this, we use the reagent ethanol. But for fats, we have to use a dry test tube and add 2 centimeter cube of ethanol into the test tube that is containing the sample. Then we have to carefully decant the water in the test tube, like in such a way that ethanol and water do not mix together. After we added the water, we will shake until emulsion of fats are produced. Fat test is positive if there is cloudy emulsion. For proteins, we use the Burette solution. We mix a crushed sample in a test tube with water and add the same volume of Burette solution, just like we did in Benedict's reagent test. We will shake the tube until there is a change in color of the solution. If the initial blue color changes to violet, then that means proteins are present in the sample. Then we have the new subtopic in our Cambridge O-level syllabus, which is deoxyribonucleic acid or DNA. These are found in chromosomes in the nucleus of the cells. Chromosomes are made up of a long, tightly coiled molecule of DNA or deoxyribonucleic acid. The DNA consists of two strands that coil together to form a double helix. Each strand consists of four types of nitrogenous bases: A, T, C, and G. The full form are adenine, thymine, cytosine, and guanine. But for O-level syllabus, we are only required to know the first initial letters A, T, C, and G. We are not required to know the full names of these nitrogenous bases. With in DNA, A binds with T with two hydrogen bonds and C binds to G via three hydrogen bonds. Again, we are not required to know the concept of hydrogen bonding in the O-level syllabus. Now we will check out some questions related to this chapter. Mostly are pretty easy, but the ones related to DNA are a bit more tough. The first question is from O Level 2023. This is a recent year question. In a strand of DNA containing 100 nucleotide pairs, 46 containing the base A. If there are 46 nucleotides contain the base A, then how many nucleotides in this DNA strand contains the base T? So how should we solve this question? If there are 46 nucleotides and for A, then there would be equal number of T in the other strand also, which is 46 here. The B would be the answer for this question. The next question is a test. The apparatus shown is used for an experiment on starch digestion. Which test tube contains the most sugar after 20 minutes? Now, we have four test tubes here. A contains starch and salivary amylase. This enzyme breaks down starch into maltose, which is a reducing sugar. B contains only starch solution. D contains starch solution and salivary amylase, just like A. The only difference between these two apparatus is that A and B are immersed in a water bath at 15 degrees Celsius. C and D are immersed in a water bath at 37 degrees Celsius. Now the question have asked after 20 minutes. Now, which contain the most sugar. So therefore, obviously we will be ignoring B and C because these are only starch solution. Now our answer lies either A or D. Now, which one would be the right answer? Since in the first apparatus the temperature is very low, therefore the rate of breakdown of starch by amylase would be very low. Therefore, there would be low concentration of sugar after 20 minutes. In the second apparatus, the temperature is relatively high, close to optimum. Therefore, the more rate of breakdown would occur for starch by salivary amylase. Therefore, there will be more concentration of reducing sugar. Therefore, D would be the right answer for this question. This question is from winter 2018 from IGCSE. Now here is a tough question. In a length of DNA, 20% of the bases were T. What is the percentage of base G in this length of DNA? Now this one is different from the first one because the first one we had A, therefore there would be an equal number of T in the other strand. But here we are given a percentage of T and it's asking the percentage of D in the other length of DNA. If there are 20% of T in one strand, therefore we will subtract the value from 100%, which is 80% that will contain the other strand. Now, in the question asks for G, therefore, since there would be an equal number of A bases in the other strand, therefore 80% minus 20% will be 60%, which is the percentage amount of both G and C bases in the entire length of DNA. 60% divided by 2 is 30, which is the percentage of base G. Therefore, the answer would be C in this question. Now, why did I do this? Because 100% represents the total percentage of bases A, T, C, and G together in the entire length of DNA. And here they have only given 20% of T. Now, see, if there are 20% of T, then there would be 20% of A in the other strand. So there will be total 40% of both A and T in the entire length of DNA. So if we subtract 40 from 100, then there would be 60. 60% represents the entire percentage of base C and G together. If we have 60%, then we obtain the value 30%, which is a value of only either C or G. Therefore, here the answer would be C. 30% percentage are base G. Now here's another question. This one is from IGCSE. A chromosome was analyzed and found to have 6,125 pairs of bases. The type of each basis was identified, and 2,345 of the bases were G. How many of the bases in this chromosome are T? Now, in order to solve this question, we first have to double the base of the value G, which is 2345, because 2,345 bases of G, if they present in one strand, then there would be an equal number of bases of C in the other. Therefore, we will double the amount. We will multiply it by 2 to obtain 4,690 bases. We will subtract this value from 6,125 to obtain the value 1,435, which is the amount of bases of A and T. Therefore, the answer here would be A, since in this entire length of DNA in the chromosome, 1,435 bases are T and A, either T and A in other strands of the DNA. Therefore, the answer here would be A. Thank you so much for attending today's session. I hope all of you have learned something. In the next session, we will be covering enzymes, which is chapter 5 of Cambridge O-Level syllabus. I hope you all of you have a great day.
Yep, so that wraps up today's episode. We hope you found it helpful and feel more confident when tackling this topic. We've got plenty more episodes coming your way, so don't forget to subscribe and hit the notification bell to stay updated. Thanks for watching, and we will see you in the next one.